This is a video on the organelle mitochondria, the singular form being mitochondrion. The mitochondrion is an organelle found in most eukaryotic cells that primarily functions to carry out ATP synthesis. That is, the process of creating the molecule adenosine triphosphate. Adenosine triphosphate, the initialism of the word being ATP, is a molecule that has three phosphate groups, a pentose sugar, which is a ribose, and a nitrogenous base, specifically adenine. The molecule is classified as a nucleoside triphosphate because of these three parts. The triphosphate coming from the three phosphate groups, and the nucleoside coming from the fact that there is a pentose and a nitrogenous base, which forms a nucleoside. This molecule stores chemical energy inside its phosphate group bonds, is a precursor to DNA and RNA, and is used as a coenzyme, which is a substance that enhances the activity of an enzyme. Inside the US education system, it is commonly called the energy currency of the cell. The mitochondrion, as shown here, has 5 parts: the outer mitochondrial membrane, the intermembrane space, which is also known as the perimitochondrial space, the inner mitochondrial membrane, the matrix, and the numerous cristae. The outer mitochondrial membrane is the outer permeable phospholipid bilayer, consisting of a 1-to-1 ratio of phospholipids and proteins. It contains porins, which lets the membrane be permeable to small things like ions and small proteins. VDAT, which regulates the flow of metabolites through the membrane, and enzymes. Additionally, the membrane can bind to the endoplasmic reticulum to form mitochondria-associated endoplasmic reticulum membrane, initialized as MAM. Next is the intermembrane-perimitochondrial space. This is the space between the outer and inner membrane. Because the outer membrane is permeable, the composition of the intermembrane space is the same with the cytosol. This is with the exception of large proteins, which must have a signaling sequence to pass through the outer membrane VDACs and other protein channels. The inner mitochondrial membrane is the non-permeable inner phospholipid bilayer that contains proteins that perform redox reactions of oxidative phosphorylation, catalyzes ATP synthesis, regulates transport of metabolites in and out of the matrix, imports protein from the ribosome, and regulates mitochondrial fusion and fission. It has a quite large 3-to-1 protein to phospholipid ratio, which translates to it housing 1/5 of the total proteins located inside the mitochondrion. Cristae, the singular form being crista, are the folds and invaginations in the inner mitochondrial membrane that increases its surface area and therefore expedites ATP synthesis. The matrix is the space within the inner mitochondrial membrane, filled with relatively viscous fluid containing mitochondrial DNA, tRNA, ribosomes, enzymes, nucleotide cofactors, and ions, which are sometimes grouped together into granules. It regulates processes crucial to ATP synthesis, such as pyruvate oxidation, beta oxidation of fatty acids, and oxidative phosphorylation. ATP synthesis, also known as cellular respiration, is the process in which ATP is formed. I'm going to focus on the animal eukaryotic cell cellular respiration, since that is the one that involves mitochondria. This process is generally divided into 3 parts: glycolysis, citric acid cycle, also known as the Krebs cycle and the tricarboxylic acid cycle, and oxidative phosphorylation, also known as the electron transport link phosphorylation, and contains the electron transport chain. With closer inspection, one can find out that the pyruvate oxidation, also known as the pyruvate decarboxylation and the link reaction, is quasi-separate from the citric acid cycle, and that other passageways exist in the eukaryotic pathway that produces acetyl-CoA, the major one being the beta oxidation of fatty acids. Now the process contains 4 steps with one extra replacement step, being the beta oxidation of fatty acids. In this video, I'm just going to outline the basics of this process. Note, the ATP, GTP, NADH, FADH2, and UQH2 yield talked about in the process is the theoretical yield. More on that at the oxidative phosphorylation step. The first is glycolysis. Glycolysis is an anaerobic process that happens here in the cytosol. It turns a glucose molecule into a pyruvate molecule and a hydrogen ion. As you can see here, it is incredibly complex. The major point here is that there are 10 steps in this pathway. The first 5 steps are what is called the preparatory or investment phase, where 2 ATP molecules are consumed. The next 5 is the payoff phase, where overall 2 ATP and 4 NADH molecules are produced. 2 reactions occur per glucose molecule. Each reaction produces 1 ATP and 2 NADH molecules. All of these reactions are catalyzed by an enzyme, and all the other processes will also have an enzyme catalyzing it. After that is pyruvate oxidation. In this step, the pyruvate produced in glycolysis combines with a molecule called coenzyme A, also known as CoA, to form acetyl-CoA, shown here. This process creates 2 NADH molecules. 1 NADH is produced by each of the 2 pyruvate that comes from the previous step, where there were 2 reactions, among other things. Slight interjection here. Among other methods, the beta oxidation of fatty acids is another way a cell can produce the acetyl-CoA required for the citric acid cycle. Here, a molecule called fatty acid acyl-CoA, consisting of a fatty acid side chain attached to other molecules, which is attached to coenzyme A, shown here, combines with a water molecule and coenzyme A to form acetyl-CoA. This produces an NADH and an FADH2 molecule. Going back to the main process, we have the citric acid cycle. This process is called a cycle since it regenerates its reactants, oxaloacetate, acetyl-CoA, and water, which allows it to run over and over again. It occurs inside the matrix for eukaryotic cells. For prokaryotic cells, it occurs in the cytosol with a slight modification, and the reaction in total produces 2 GTP-ATP. Guanosine triphosphate has the energy equivalent of adenosine triphosphate because they have the same structure except the nitrogen space. 6 NADH and 2 UQH2, also known as ubiquinol. This is from the 2 full runs of the cycle, each one producing 1 GTP-ATP, 3 NADH, and 1 UQH2. The name comes from the citrate formed in the beginning steps, which is derivative of citric acid, which makes it the citric acid cycle. The final step in synthesis is oxidative phosphorylation. Here, the electron transport chain, initialized as ETC, exists. The ETC releases the energy of NADH and UQH2 through 4-5 protein complexes. The first 4 creates a proton gradient between the intermembrane space and the matrix. The final complex, called ATP synthase, uses the potential energy stored in the proton gradient to attach a phosphate group to an adenosine diphosphate molecule to create adenosine triphosphate. There are an abundance of other ETC processes, but this is the most efficient one used by mammals. In total, the theoretical yield for oxidative phosphorylation is 34 ATP molecules. This is just the release of the energy from the product of the previous step, which allows it to create ATP. And the total theoretical yield for the entire ATP synthesis process, without the beta oxidation of fatty acids, is 38 ATP molecules, assuming a 3 ATP per NADH and 2 ATP per UQH2 conversion. This is because glycolysis produces 2 ATP and 4 NADH and consumes 2 ATP molecules. Then the pyruvate oxidation produces 2 NADH, and the citric acid cycle produces 2 GTP-ATP, 4 NADH, and 2 UQH2, yielding a total of 4 ATP, including the 2 GTP, 10 NADH, and 2 UQH2, so 38 ATP molecules. However, in reality, things aren't as efficient. During the process, 2 NADH molecules are transported into the matrix through the energy given off by 2 ATP molecules, and leakages in the proton gradient makes the ETC less efficient. Researcher Peter Reich from the Department of Biology at University College London estimates an approximately 29.85 ATP molecules, rounded up to 30, yield with an approximate 2.5 ATP per NADH and approximately 1.5 per UQH2 yield in his 2003 paper. Sorry if I pronounced his name wrong. In summary, the mitochondrion is an organelle inside eukaryotic cells that mainly functions to carry out ATP synthesis. The organelle has 5 parts: the outer mitochondrial membrane, the intermembrane space, also known as the perimitochondrial space, the inner mitochondrial membrane, the matrix, and numerous cristae. ATP synthesis has 4 major parts: glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, with the beta oxidation of fatty acids as a step that can replace pyruvate oxidation and glycolysis. Without the mitochondria, the cells would not be able to carry out activities, since they would have no way of reliably transferring and getting energy.